In this activity is called protein bracelets, and it's a great way to learn about mutations. We'll begin with the background. Please make sure you're following along in your document as I read aloud. What are mutations? Mutations are changes in the DNA strand. They may be beneficial, neutral, or harmful for the organism. Mutations do not try to supply what the organism needs. Instead, they are random, meaning that whether a mutation happens or not is unrelated to how useful it is. Mutations occur naturally during DNA replication. However, harmful chemicals such as tobacco, ultraviolet radiation, and X-rays may also lead to mutations. There are several types of mutations. Insertions and deletions are additions and losses of nucleotides on the DNA strand. These two mutations are referred to as frameshift mutations because they lead to a change or shift in the mRNA sequence of codons. A point mutation or substitution is a change or substitution in a single nucleotide. There are two types of point mutations: missense and silent mutations. Missense mutations lead to changes in a single amino acid, whereas silent mutations have no effect on the amino acid sequence. Let's try to digest just that much. First of all, mutations are simply changes in the DNA sequence. We're really talking about the nitrogenous bases here. There are two types of mutations. There's frameshift and there are point mutations, also called substitutions. Frameshift mutations come in two flavors: insertion, where a nucleotide is added, and deletions, where a nucleotide is taken away. Point mutations or substitutions also come in two flavors. There's silent mutations, which mean that a nucleotide is changed, but it doesn't have any effect on the resulting protein, and missense mutations, which do lead to a change in an amino acid in the protein. Below our background reading, we see a color chart. We'll need to use this when it comes time to build our protein. For now, let's move on to the procedure. According to the procedure, step 1 says, transcribe the DNA sequence into an mRNA molecule. Write the complementary bases beneath the DNA sequence, A, U, C, or G. Translate the mRNA codons into a sequence of amino acids using the codon chart in your textbook. Remember, this is Chapter 14, Lesson 2. Write the name of the amino acid coded by each codon in the mRNA strand. You may use the first 3 letters of the amino acid. Use markers to create the resulting polypeptide composed of amino acids. To do this, simply color in each circle to represent an amino acid. Use the color chart above to help you. For the amino acid serine, just leave the circle blank to represent the color white. Answer the questions that follow in complete and descriptive sentences using scientific language. Let's look at our standard DNA sequence. We need to transcribe the DNA sequence into the mRNA complementary strand. TAC will transcribe into AUG. You'll have to forgive my poor writing here. I'll try again. AUG. The next sequence, GAA, will be transcribed into CUU. AGA transcribes into UCU. TGA transcribes into ACU. GAG transcribes into CUC, and so on. You'll have to forgive my sloppy handwriting, but here I have transcribed the first 7 DNA triplets into the 7 mRNA codons. Now I have to use my codon chart to then translate the codons into amino acids. Using the codon chart, AUG codes for MET, CUU codes for LU, UCU codes for SEER, ACU codes for, we'll just put THR here for short, CUC codes for LU again, UCA codes for SEER again, and ACG codes for the THR. Using the color chart at the start of our activity, we can now use markers to color in our amino acids. Methionine is colored yellow, leucine, red, serine, white, so we'll leave that one blank. The THR, we're going to color blue. Now we're back to leucine, red, serine, white, and then the THR blue again. And you're going to see that as you keep transcribing and then translating that DNA sequence, we'll end up with a pattern that is quite patriotic. Now, we move on to the analysis questions. What is special about that first codon? Well, the first codon, AUG, stands for the amino acid, or codes for the amino acid methionine. But methionine serves as a start codon. What we mean here is that this indicates the start of transcribing the DNA molecule into an mRNA strand. Number 2, why does the sequence of colors or amino acids repeat? Notice that we have red, white, blue, red, white, blue, red, white, blue. However, if you look up above in the mRNA sequence, even though we have the same colors, we don't necessarily have the same codons. And so, why might this be? This is because if you look at your codon chart, there's only 20 amino acids, but there's over 60 codons. 60 plus codons? Yet, only 20 amino acids. What this means is we have redundancy in the DNA code. That's a good English term right there. What we mean is there's a lot of codons that code for the same amino acid. Let's move on now to our first mutation. This is called a same-sense or silent mutation. You'll see why towards the end. It says to mutate this DNA sequence, change the second A in GAA below to a T. Rewrite the entire DNA sequence under mutated DNA sequence. Then transcribe and translate this new sequence and answer the questions. So, we are simply going to rewrite the DNA sequence, but do exactly what it says. We're going to recopy the DNA sequence, but when we get to that A right here, we are going to change it into a T, just like it says. And then we're going to keep copying the code, and so on. Now let's look at the mRNA sequence. You will transcribe the sequence just like you did before with complementary base pairing. But notice when we get to this second triplet, we have C, U, A, rather than the original C, U, U. And then we keep going and transcribing the rest of the sequence. Now let's translate the mRNA code into amino acids. AUG codes for MET. That hasn't changed. But what about this codon now that it has a changed nucleotide or substituted nucleotide? Will it still code for the same amino acid? Look on your codon chart and find out. Once you determine the amino acid, I think you'll understand why it's called a silent mutation. Remember to use the color chart then to color in your amino acids. This will enable you to see whether or not the mutation has changed the resulting protein or not. So, what do you think? Me coloring it in, that's your big hint there. The next mutation, missense mutation, should lead to a change in the amino acid. Let's start by rewriting the mutated DNA sequence. At the top, remember it says to mutate the sequence, change the first A in the third codon to a G. So here we go. T, A, C, G, A, A. Now I have to change that first A into a G and then continue writing the DNA sequence. Now let's look at transcribing the DNA into the mRNA strand. TAC transcribes into AUG, GAA transcribes into CUU. But now that we've changed that first nucleotide, we've made a substitution. We now have the codon CCU. And then keep transcribing the rest of the DNA strand. When you move on to translate the codons into amino acids, be really careful here. AUG still codes for MET. CUU codes for Lu. But what about this new codon? Now that we have a substitution here, will it code for the same amino acid? Look at your codon chart to figure it out. Make sure you look at the color chart to color in your amino acids using the proper colors, and then answer your analysis questions. We're now moving on to our frameshift mutations. Remember that these are ones where we're adding or subtracting nucleotides on the DNA molecule. The first one we'll study is insertion mutations. It reads, to mutate the sequence, add one base to the gene by adding a G between the TGC and the GAC DNA triplets. Okay, this is a little more complicated here. So to begin with, you will just rewrite the DNA strand as you see it above. However, once you get down here with this insertion, you're going to see a shift in the triplet sequence. We have T, G, C, but now we have G inserted, followed by G, A, followed by C, A, G, followed by G, T, G, and of course you just have this lonely little T hanging off at the end. Don't worry about that little T. You can ignore it for now. But what you really need to see is that because of this insertion, there has been a shift downstream. Here I've rewritten the mutated DNA sequence from this section on below. Now, when you go to transcribe this DNA sequence into the mRNA codons, we now have 1, 2, 3 different codons after that point of insertion due to this shift. Notice how this is going to then affect the 4 amino acids at the end of your polypeptide. How many color changes are you going to see? Answer the analysis questions that follow. Now for our final mutation, deletions. These are usually the most harmful form of mutations in humans. You'll see why in a moment. The directions say delete one base from the gene by dropping the second G in the GAG DNA triplet. So what we want to do is get rid of that nucleotide. That means that all of these nucleotides are going to shift upstream, changing the mRNA sequence. Let's start first by rewriting the DNA sequence. Here you will simply copy all the nucleotide base letters, and then when you get here, you want to make sure that you delete that G and show that shift. So here we have G, A, G, T, T, G, C, G, A, C, A, and so on. Then you will transcribe those nucleotide triplets on the DNA strand into your mRNA sequence. You should notice a change in mRNA codons after that deletion. And then you should see a change in the amino acids that those codons code for. In other words, really pay attention to what's happening towards the end of your polypeptide. And then again, answer the analysis questions. Take some time to answer each of the 5 conclusion questions. Remember, there's an answer key on the portal. If you don't understand how to answer these, and you've checked the answer key and still don't understand, make sure you contact me. That applies to this matching exercise. So let's review here. Our unmutated standard DNA sequence produced this polypeptide or protein made up of 10 amino acids. Then we tried 2 different substitutions, a silent mutation and a missense mutation. In both types of mutations, all we're doing is substituting one nucleotide on the DNA molecule. But look at the difference between the two resulting proteins. In a silent mutation, the resulting protein is unaffected or unchanged. Yet, in a missense, you can see that where that one DNA nucleotide was changed, it resulted in the change in one amino acid in our polypeptide. Now let's look at our two types of frameshift mutations, insertion and deletion. This is where we either add one DNA nucleotide or we delete one DNA nucleotide from the original DNA molecule. The resulting protein has some major changes to it. I can see right here that this was the point of the insertion on the DNA molecule, because now I have 3 new amino acids in my protein. Deletions tend to be worse, and you can probably see why. Instead of having 10 amino acids, we only have 9 amino acids. And right here I can see where the mutation occurred. I now have 4 amino acids that are new in my protein. Now, how will these proteins function? We're not sure. Like the intro said, sometimes these proteins could be beneficial, or they might be really harmful, or they may really cause no effect on the organism at all. It just depends on the mutation, and it just depends on the protein that the DNA codes for.